All right. Fun drive. Okay, so let's go over what. So first thing is that we covered a little bit from chapter 14, which about amines, and amines are ammonia. So ammonia is NH3. It's ammonia with carbons attached. So if we replace the hydrogens with carbons, we get something like that. Okay, and we're going to have three different types of these. We have one carbon attached to the nitrogen. It's called primary amine. We have two carbons attached. And I'm going to just draw a couple so we can name them as well. And we have three carbons attached. It's called tertiary. All right, so what we want to do is be able to identify if you have a primary or tertiary or a secondary, and also name them. Okay, so this is the primary. It has a methyl attached to it. So it's methylamine. Okay. This has two carbons attached to the secondary. So the name, and the name of this one is going to be ethyl methyl amine. And this nitrogen has three carbons attached. So it's going to be, has two ethyls, so it's diethyl. It's a tertiary, so it's diethyl. Methyl amine. Okay, so that's what all you have to know as far as the amine goes, as far as naming and identify what type they are. Okay, one of the reasons why we're covering these is because these are found in proteins. There's one particular reaction that we're going to refer to, and when we get to proteins, that involves amines. And it's called amidification, and it's a lot like esterification. So I'm going to write down the esterification reaction as well, just to parallel them. Okay, so esterification. All right, this way you take an alcohol with a carboxylic acid, and you end up with an ester and water. Okay, so for example, and we're not going to worry about naming these, okay, you won't have to give the name, but let's say you had ethyl alcohol and this guy here. So what you're going to have to do is you're going to have to snap off the water. So we're going to snap off those two guys. And then you combine these two parts. And then you have your ethyl. And then you have your and water. And I'll call this is an ester bond. Okay. Now, be able to do this. It might be on the test, okay? Even though it was for last test, I want you to be able to parallel the two type of reactions. Okay, so the other one is the one that we're going to be using for proteins is amidification. and that's where you're taking a mean. In this case, I have a secondary. You have to have a hydrogen. So tertiary amines will not perform this reaction because they don't have a hydrogen that pops off. And you won't have to name these guys, you just have to, may have to complete the reactions. And so what happens is you get pop-up water, and this does it a little bit different here. The OH came from here instead of over here, but this. Don't forget the water. All right. So what you do is you make an amide, and what holds it together is an amide bond. Okay. So that's all you have to know about amines right now. But we're going to apply it to proteins. It's key. From my computer. Okay, so now we're going to take this information and other information that we've learned and give them proteins. So proteins are a biological macromolecule. We've covered carbohydrates, where the building blocks are monosaccharides, and you can make starch and you can make glycogen from them. We talked about lipids, and the building blocks for lipids are fatty acids. For proteins, the building blocks are amino acids. And so what you're going to do, we're going to learn about them. So we talked about the amino acids individually, and then what you do is you link the amino acids together to make proteins. Okay, so by the way, thank you for trying to get in today. It was a little scary. I walked out and my driveway was cheap lice, and because I live out in the country, and trees were just.
When I walked in, it was branches were falling.
Yeah, but once I got on the road, it was okay. So I had. So I wore a helmet just in case. And I had those things that you attach to your shoes, tracks they're called, or so I wouldn't fall down. They're great though. They're really good. Okay. So I am taping this, and I will post it either today or tomorrow. Hopefully it'll come out okay. And this can help you with review if you need it. And there might be an outline. If not, I've redone the PowerPoint. I might be just sending you the PowerPoint. Okay, so chapter 16 is proteins and enzymes, and show you what you need to know. Okay. All right. So proteins are made out of four elements: carbon, hydrogen, nitrogen, CHON, C-H-O-N, and sulfur. And you don't really have to know this, but because if you look at all the amino acids, you'll notice that these are here. What makes these different than fatty acids and carbohydrates is they have nitrogen attached to them. The carbohydrates and fatty acids just had carbon, hydrogen, and oxygen. And a couple of amino acids have sulfur in them. We don't talk about sulfur a lot in this class, but there's a really important reason why sulfur is found in the body. One real important part is sulfur is really good at reducing things. And so what is it? It's an antioxidant. So your body has natural antioxidants, and one of them is called glutathione. It may be the most powerful antioxidant that there is. And what it does, it has a couple of sulfurs on it, and the sulfurs go around and look for oxygens and electrons that are flowing or that shouldn't be there, and scavenge them up. So we don't talk about sulfur a lot, but it has a really important job in our systems. Okay. All right. So the list of the functions. So we have carriers, which are hemoglobin and lipoprotein and others. All right. Cell wall transport. You had the active transport and facilitated for moving things in and out of the cell, because most things are too big to go in the cell, and so they need help. Then you have muscle movement, and that would include all organisms usually have to have some way of moving. I don't think amoeba have a way of moving except for just sort of, diffusing. But most things need some sort of way to propel. And we have muscles and there's flagella like on sperm, and cilia found on, you've seen this stuff, I'm sure, in your biology classes. There's antibodies which defend against foreign proteins. So if you get a virus, or if some sort of allergen gets in your body will produce an antibody against it, so that, because it says it's dangerous, let's get rid of it, sends it to the T cells. Structure, your connective tissue, hair, fingernails. The cell also has its own structure, and so when you go inside, there's all kinds of little structures that we don't even talk about. All right, and that you wouldn't even know if you weren't in this class. You can just be ignorant to them. Okay. Hormones. Most proteins are protein hormones like insulin, glucagon, pituitary, thyroid, stimulating, and so forth. And what they do is they signal. They tell one part of the body that something else is taking place in their body, and we have to be aware of it and keep it under homeostasis. You have cell receptors, which are found on the cell, and which grab a hold of the signals, and other parts like neuroreceptors, and then you have enzymes, which we're going to spend at the end of this. Then finally there's food. And I may have left one out, but this is good. Because I want to know to let you know that proteins are more than just hamburger in building muscle. They're almost everything that gets done in your body, and what you're held together with is protein. I mean, it's the most important. I mean, everything is important, but there's so much importance to these guys. As far as food goes, really, you eat protein to replenish the amino acids, because these guys are falling apart all the time, and they need to be rebuilt. Now, if for some reason you go into complete starvation, your body will break down. If it runs out of carbohydrates and runs out of fats, it'll go and start breaking down your protein as well. Normally, also, there is some protein that's being broken down during the day, but it's really low on the totem pole. All right, so the building blocks of proteins are amino acids, and we'll show how to make those how proteins are made up in a little bit. and they're connected by peptide bonds, we'll show. This is sort of getting you ready for what we're gonna talk about. There's 20 amino acids, common ones, there's more than 20, but we only refer to 20, and you know, it's to give you a starting point. And, okay, that's good. So, let's see. All right, so this is what, this is an amino acid, okay? It has, here's an amine. Okay, so it's a ammonia with a carbon attached to it. And it's also a carboxylic acid. So it's both amine and a carboxylic acid. Amines are bases in chemistry. So amino acids are kind of interesting because they are both a base and an acid. They have a base part and acid part. The R represents the side chain or side group, and up 1 to 20 amino acids. Each amino acid will have this in common. This guy is called the alpha carbon. But coming off the alpha carbon will be an R group or side group, which is different for every one of the amino acids. I didn't, and so I told you we went over to 20 and their names. Okay, but we're in, you said, well, maybe I have to memorize them. No, okay. But what we need to know is what properties do we want to know about them? What's important to know about their structure? Because their structure will lead to how they, how they show up in proteins when we're all done. Okay, so we have proteins. So 20 proteins can be broken up into. I've had, make sure it's going to be a little bit different than this, because I'm taking a couple of them out. They have the nonpolar. Okay, what makes the nonpolar is the carbons and the side chain. Because the more carbon you have, I told you, it's going to likely be more nonpolar, hydrophobic. Okay. The other thing is that there are 9 of them, okay? And that's kind of important when we show the ratio of the other ones. Just some things about these is that these side chains are actually kind of big, and they're long and they're kind of big, and they can kind of squiggle around a little bit. These are what the bodybuilders would call branched chain amino acids, the BCAAs. And we'll talk a little bit more about things. Okay, so there's 9 of those. Okay, there's one that looks a little bit different. What it does is it kind of goes back beyond itself, but it's called proline, and that's a really interesting amino acid. So, kind of cool thing happened yesterday. I was just playing around at some time, and I looked up. So my PhD thesis was to predict the structure of a protein, and I did it back in 1987. And I was just curious, and if somebody had actually came up with the actual structure, and they have, and it was really cool. So I compared my structure against a real one, and it works really good. And so that was over 30 years ago that I predicted this structure, and it's almost very, very similar to the one that's predicted. And that's because I know a lot about amino acids. So, and I go, oh, I may say this guy's interesting. You guys don't know what that means. But when I think about it, I go, oh, I know what that means. And that's why when I looked at the structure, I go, oh. And so it's, I don't know, you kind of see a different world when you get into these things. All right, okay. All right, polars. All right, polars, what they have are oxygens in their side groups. Okay, you'll notice is that all of these have oxygens, and we're gonna take this guy out. It has a sulfur in it. And I told you what things, sulfur is important. All right. That's almost, it's so important that we have to pull it out, put it in its own special group. Okay, that's what we're gonna do. So, if we pull that out, what we have is we have 1, 2, 3, 4, 5. We have 5 of them. Okay, and then there's acid base, and there's 2 of each. Okay, and, all right, and then finally, the last one, we're gonna take out histidine, and we're gonna put it as a special. So we have 2 specials, we have cysteine and histidine. Okay, now, all right, so now it's just not random that there is more nonpolar amino acids than anything else. Okay, when we look at a protein, and we'll talk about how this gets formed in a minute. So when a protein is formed, and this we'll get to the how this is formed in a minute. But when a protein comes off of DNA, so when DNA makes a protein, we'll talk about that in the next section. It comes off as a long chain of amino acids, and that long chain of amino acids is mostly nonpolar amino acids because there's more nonpolar amino acids than anything. And so what they do is they see the water, and the water kind of pushes them in, and so pushes them in together, and so they form a sphere like soap. So like when you form soap, the tails go inside, and then the polars go on the outside. Well, you get a similar thing here. You get the nonpolar guys go in the middle, and the polars go on the outside. Well, if you look at the difference between the inside and the outside, there's more inside than there's outside. So you need to have more nonpolar than you need to have polar. Okay? So more inside than outside. I hope you see what I'm saying. There's more volume than surface area. So if, like I said, is if you take duct tape and you put it around a basketball and then you put it in a basketball, you're gonna have a lot more tape inside the basketball than you are on the outside of the basketball. Okay, so you need to have more nonpolar than you do polar because the polars are gonna be on the inside. Okay. So that sort of explains why those guys are there. These guys over here, the acid base and the special, are found being mostly with function. These guys here are involved in building the structure. And that's really, a protein is mostly just structure and not really function. The structure is there to build a really kind of small section which is going to do the work. And when we get into like enzymes, this is the workspace, work area, whatever, which we'll call the active site or binding site, and it's really small compared to the rest of the protein. And the reason why that is because it's very specific. It has to be you have to move all these big things around to get this just right, okay? It can't be willy-nilly, just throw a couple things in there, it's gotta be pretty precise. So these guys are involved in building big things. While these guys over here, the acid and base and the specials are really more involved with the function. Now there's crossover. There's some of those guys are found in function, some of these guys are found in the structure, but it's pretty good if you look at a protein and you look for these guys, if you look for these guys in the protein, you probably can find the work area. Okay. So. So we're going to talk about how all this is formed together. The key things is that most of a protein is non-polar. Now there are times when some proteins are mostly polar. And that's fine because they take advantage of their environment to be that way. I don't know the protein structures, but I could spend a whole semester or two talking about it, and maybe I will do that today. Maybe I'll just make this class on. All right. Okay. Next. Kill me. Just gotta go with the flow here. Any questions so far? Almost have a full house. What's up with that? What day was it, on Tuesday? We're only like 4 people here. Oh yeah, so how many of you went to nursing meetings? How about at the 8 o'clock session? So, yeah, thanks for sending me that. I guess there was a couple other sessions, like at 10 or something, 10 to noon. It really wasn't that big of a deal in terms of attendance. But the thing is you guys kind of missed out on, and there's stuff that I'm just gonna kind of run through today, and that's what I'm sad about. All right, so briefly, we will talk a little bit about the nutritional value of proteins. Now, the reason why you need protein in your diet, like I said, is not really for energy. It's to rebuild the proteins that fall apart. Because proteins are constantly falling apart, just like everything, you know. It's just, your muscles are falling apart, your brain is falling apart, you know, everything. Really notice it more as you get a little bit older. Your hair starts falling out, your teeth start falling out, they just start falling apart. Okay, so you got to replenish those amino acids. There's some of the amino acids your body naturally makes. It says, oh, don't worry about it, you guys do just, I'll take care of it. But there's some that you need to get in your diet, and those are called essential amino acids. And depending on if you're a child or an adult, the number varies. There's 10 if you're a child, 8 if you're an adult. Now, one of the things that I brought up that to me, it's really kind of interesting, kind of cool, is that if you look at the essential amino acids, of the eight that adults need, 6 of them are non-polar. Okay. All right, and then I think to myself, I go, wow, that's really interesting. What is the most abundant type of amino acid out there? Non-polar. Okay. So really, in a sense, to me, these really aren't essential because you're going to find them in just about everything you eat. You know, you don't have to worry about eating valine. I mean, it's going to be there because the most abundant type of amino acid is protein. So if you just eat food, you're probably going to get the amino acids. You're going to get these amino acids. All right. Okay. It's just interesting how nature takes care of you. It just says, don't worry about it. Just eat. All right. And things will work out. Okay, there's complete protein foods and incomplete protein foods. Complete, and that's sort of, it's worded kind of wrong. It says a protein that provides necessary, it should be a food. So you're not going to go, oh, I'm going to eat trypsin or carboxypeptidase. You're going to eat a hamburger, okay? So, and we'll see in the next slide about that. All right, so here's kind of a summary of different foods and if they're incomplete or complete. At the top, eggs, milk, meat, fish, and poultry are all complete proteins. They all have the requisite essential amino acids, and the other ones you can see don't. And you know, again, this is curious to me because it turns out is that, you know, let's say you have, say, beans. If you're eating beans, what should you eat with beans? Rice and beans, okay? That's kind of a staple around the world, is that kind of meal. So we don't have to worry about finding which essential foods, amino acids, because they're probably going to be in our diet, except our diet. Because sometimes we specialize our diet so much by going to McDonald's. Okay. And also this, I've read recently that maybe this is not all that it should be, that every food has the essential amino acids in it. And we should re-look at this. But for now, let's just stick with that. Okay. All right, so what we want to do is talk about how proteins are made. And the first thing we need to do is make a peptide. A peptide is 2 amino acids together, all right? So